and I'll let you take it away.
Great, thanks so much. Thanks for organizing this. I know it's no small feat. Okay, we can see the screen, right?
Yeah, we can see it.
Okay, sorry, I lost you all. No, okay, so I'm just gonna talk as if you can hear me. Thanks for organizing. Especially during a pandemic, I know it's not easy to corral a lot of people, but this has been great so far. I'm really excited to share the work that we're doing in my lab. Focused on neuroinflammation in the context of viral infection of the central nervous system, the brain and spinal cord. So in this talk, I'm going to sort of start with, a couple of stories that I publish as a postdoc, and then kind of transition into the ways that we're following up on those stories now in my lab here at Rutgers. We're really interested in a cell biological phenomenon known as programmed cell death. As many of you no doubt have seen, in courses, in your training so far, almost everyone is familiar with apoptosis. This is a form of programmed cell death in which cells sort of swell and bleb, and they end up in these self-contained membrane-bound vesicles called apoptotic bodies. These apoptotic bodies are cleared very effectively by phagocytes of the immune system, and for that reason, we think of it traditionally as a non-inflammatory form of cell death. They get cleared, and then that's sort of the end of the story. Apoptosis is coordinated by a family of molecules known as caspases. In contrast, we've come to understand, I would say over the last 15 years or so, that there are other types of programmed cell death beyond apoptosis. One that's relevant for my lab's work is called necroptosis. Necroptosis looks like accidental cell death or necrosis, when a cell loses its membrane integrity and kind of pops and releases its cytoplasm into the extracellular environment. It looks like necroptosis, but it happens on purpose. So it sort of has this combined name of necrosis and apoptosis, and that's where we get necroptosis. Necroptosis, in contrast to apoptosis, is highly inflammatory. And that's because the cytoplasm is full of damage-associated molecular patterns and other immunogenic stimuli, which induce a really potent inflammatory response in the tissue. And this process is mediated by a family of molecules known as the RIP kinases, receptor interacting protein kinases, although I always joke it's a little more fun to think of them as rest in peace kinases. But regardless, this form of cell death, which is a lot more recently described, we now know is really important for host control of a number of different bacterial and viral infections. I want to dig a little bit deeper into kind of the main molecular players before we really jump into it, though. And this is going to be pretty simple, so we'll keep it to the main players here. But what I want to say is that necroptosis is triggered downstream of immunologic receptors. So this could be a death receptor like the TNF-alpha receptor or a pattern recognition receptor. Regardless, ligation of one of these receptors activates the first molecule in this cascade known as RIP1, RIP kinase 1. RIP1, sort of in the most simple version of this pathway, recruits RIP3, which is kind of the central signaling molecule, the one that we'll focus on the most. When RIP3 is activated, it recruits additional copies of RIP3 and sort of forms this oligomeric signaling platform with RIP1 that goes into the cytoplasm where it's free to phosphorylate its targets. Now, its main target is a single molecule known as MLKL. And so MLKL, what's important about it is that it is the executioner protein of necroptosis. So when MLKL becomes activated, it targets itself to the host cell membrane. It forms this oligomeric pore structure, like so, and this pore allows water to rush into the cell. When this happens, the cell swells and ruptures, and this is what is the sort of proximal cause of cell death. And when the cell ruptures, again, it releases its contents into the extracellular environment. So this is sort of also the source of a lot of the immunogenicity of necroptosis. So when I started my postdoc, necroptosis was still pretty new. It was not brand new, but had not been really thoroughly characterized in terms of roles for this form of cell death in host defense against pathogens in vivo. A lot of the molecular signaling had been worked out. But when I started my postdoc, I was really interested in seeing whether this form of programmed cell death was important for the control of a family of viral infections that I've had a long-standing interest in since my time as a graduate student. And these are flaviviruses. So many of you probably recognize at least a couple of flaviviruses. And for the purposes of this talk, I'm going to talk about West Nile virus and Zika virus. The main thing to know here is that These are insect-borne viruses. These two in particular are borne by mosquitoes. And why they're relevant to me as a neuroimmunologist is that they're very good at accessing the central nervous system, establishing infection primarily in neurons, and in many cases causing a potentially fatal encephalitis. This is a West Nile virus in particular. It was introduced in 1999 into the United States, but of course it's all over the globe. It causes, it's the leading cause of epidemic viral encephalitis in several countries. And then Zika, which sort of emerged more recently, at least in the Western hemisphere, has sort of similar symptoms, but of course, it's able to do some of these effects in utero actually. And so it has attracted a lot of attention for that reason. Things that most flaviviruses, or at least the sort of brain relevant flaviviruses have in common is that they're highly neurotropic, meaning that they infect neurons and they productively infect neurons at really high rates. And so we see this across a mammalian species. So for example, we can see infected neurons in the cerebral cortex of a mouse here, really widespread cortical infection. And then these are infected neurons in the cortex of a human patient who'd actually died of West Nile virus encephalitis. Once infection progresses to this point, typically it is fatal. However, this is not what always happens. Many times flavivirus encephalitis can be self-limiting. And this is due to the innate immune function of neurons and other resident cell types of the brain. So resident innate immunity of brain cells is very important in the context of flavivirus infection and other viral infections of the nervous system. But typically, the cell intrinsic innate immune response is not enough. We also need to recruit immune cells from the periphery, particularly adaptive immune cells, to really fully fight infection and ensure survival of the host. However, in the brain, this is a little bit challenging because while we need to recruit T cells, here marked in red, two sites of antigen burden in the brain, so we can kind of see that here. We're recruiting T cells to a population of infected cells in the brain. We need that to happen to clear infection. If we don't have a T cell compartment, mice uniformly succumb to infection. While that is beneficial, that process also can be somewhat of a double-edged sword because the recruitment of T cells into the brain causes a lot of bystander injury and immunopathology. So I've showed some of this in my work previously. Here you can see sort of a really inflamed mouse brain, all these T cells marked in green, around a whole bunch of tunnel positive dead cells in red. Most of these dead cells were probably not infected. Understanding the molecular mechanisms that promote protective neuroinflammation versus pathologic neuroinflammation is really one of the key interests of my lab. And of this field generally, I would say. And so how do we understand those molecular mechanisms? Well, we have a few tools to go about that. We have these really nice, robust mouse models of flavivirus infection, for example. So we can do a subcutaneous inoculation of these viruses into a rear foot pad. This mimics a physiologic route of infection from a mosquito bite, for example. The virus would come in under your skin. Both of these viruses, Zika and West Nile, they disseminate through the secondary lymphoid organs and can access the central nervous system about a week after infection, where depending on the system you're using, they can establish a potentially fatal encephalitis. So this is nice when we want to study kind of natural infection, natural immune priming in the periphery and that sort of thing. However, we can also inoculate these viruses directly into the brain using an intracranial route. And this is nice when we want to ask a more targeted question about local innate immune responses, or we want to control for some of the confounds of a peripheral infection. You have different levels of immune priming, you have different timing of neuroinvasion and different anatomical location of neuroinvasion in the peripheral route. So the intracranial system also helps us ask different questions. And so we'll use both of these systems in the data that I'm going to show you today. So going back to the beginning, putting these pieces together, I was really interested in figuring out whether or not necroptosis, this non-apoptotic form of programmed cell death, was it important for contributing to the host response to flavivirus encephalitis? And so I did this in the course of two different papers. I'm kind of going to hit the highlights of them here. But just starting with kind of one of the very first experiments I did, which is just to take mice that are lacking various components of the necroptotic signaling pathway. So RIP1, RIP3, or MLKL, infect them subcutaneously in the foot with West Nile virus and just sort of see if they survive that challenge. And what we saw is that mice lacking RIP3, that kind of central signaling kinase in the pathway, they absolutely required RIP3 to survive challenge with West Nile virus. You can see they start to die faster and they universally succumb to that infection. We saw very similar results with mice that were lacking the kinase activity of RIP1 for complicated reasons. We don't use knockouts of these animals. But these are functionally dead RIP1 mice, and they also succumb to infection. So we need RIP1 and RIP3. However, when we did these experiments in mice lacking MLKL, which remember is the executioner molecule of necroptosis, these mice were indistinguishable from wild types. And so this suggested to us that while RIP1 and RIP3 seem to be absolutely required for survival from West Nile virus, MLKL was not. And so it seemed that RIP kinase signaling was acting through a mechanism that was independent of its canonical function in mediating MLKL-dependent necroptosis. In a follow-up paper, we saw basically the exact same sort of phenotype after infection with intracranial Zika virus. And so if we inoculate all three of these transgenic lines with Zika virus directly into the brain, we see that mice lacking either RIP1 or RIP3 are very susceptible to that infection, while MLKL knockouts were not. And so going forward, you can kind of assume that every experiment I showed you, I did an MLKL knockouts and nothing happened. I've never seen any MLKL dependent phenotype in a flavivirus infection model. However, RIP kinase signaling was required. So if RIP kinase signaling is not acting through MLKL, its canonical substrate, of course, we want to know, like, well, what is it doing instead? If it's not necroptosis, what's happening? Well, to figure that out, we really needed to sort of understand why the mice were dying in the first place. And I won't show all the data, but we saw that mice lacking RIP3 in particular were perfectly capable of mounting a normal systemic immune response. They could prime antigen-specific T-cell responses. They formed neutralizing antibodies. And in fact, they cleared virus effectively in the periphery. And by the periphery, I mean outside of the nervous system. So, you know, in the secondary lymphoid organs, in the serum, they look pretty normal for the most part. However, when we turned our attention to the central nervous system, we saw that instead, mice lacking RIP3 had increased titers of West Nile virus. This is the West Nile virus model again, across the brain, across the central nervous system, actually, getting sort of progressively worse until the terminal time point. So mice begin to die around this point. And you can see they have significantly higher viral burden in the cerebral cortex, but also in other brain areas like the brainstem, the cerebellum, and then finally the spinal cord, where the virus sort of gets to last in this model. So universally across the nervous system, RIP3 mice failed to control viral replication within the nervous system. We saw, again, a similar phenotype in the Zika virus challenge model. This is with intracranial infection now, again, with Zika virus. But you can see a greatly increased viral burden in the brains of RIP3 knockout mice, RIP1 kinase dead mice, but again, not MLKL knockout mice. So MLKL was dispensable for this effect. So, you know, we did a lot of work to sort of characterize what the mechanisms were that promoted this failure of virologic control. It ended up being slightly different between West Nile virus and Zika virus. I'm going to focus actually on the West Nile virus story for this talk, just because I think it's a little more interesting to immunologists probably. But there were both sort of cell intrinsic and cell extrinsic mechanisms of this effect. And so RIP kinase activity in neurons themselves had some antiviral activity, particularly for Zika virus. But what was really interesting is that when we really dug into how RIP kinase signaling, particularly in neurons, controls the overall pathogenesis of infection, what we saw was that RIP kinase signaling in neurons actually promotes transcriptional activity of a number of antiviral and inflammatory genes. And one sort of population of genes that ended up being very important were molecules known as inflammatory chemokines. So for those of you who are not familiar, chemokines are molecules of the immune system which are really important for inducing the migration of immune cells, and in particular are really important for recruiting immune cells to sites of infection. So when you have an infection, particularly in the brain, infected cells in the brain begin to express these chemokines which kind of draw immune cells out of the circulation and into the brain in order to access antigen and go about the work of clearing infection. What we saw was that mice lacking RIP3, or in this case, these were actually neurons that we cultured in a dish in vitro and infected with West Nile virus. And we saw that they lacked expression, or they had diminished expression of a number of really important inflammatory chemokines. This included CCL2 and CXCL10, which I'll explain in just a minute. But we saw this both at the secreted level in the protein and the supernatant, but we also saw it at the transcriptional level. So this effect was happening at the level of mRNA. I've sort of, there was a bunch of chemokines that were different in our assays, but I focused on these two because we know, particularly for West Nile virus, these two chemokines are absolutely essential for the promotion of beneficial neuroinflammation and ultimately survival of the host. And so what are these two chemokines? Well, CCL2 is important for recruiting a population of myeloid cells, particularly monocytes, which express the cognate receptor CCR2. So if neurons are expressing CCL2, that's going to attract CCR2 expressing monocytes to the site of infection. Similarly, CXCL10 is pretty important for recruiting T cells. And these T cells recruit the CXCL10 recruits T cells which express its cognate receptor CXCR3. So a little bit of alphabet soup going on here, but the idea is if we have less of these chemokines, we should be recruiting fewer of the immune cells that recruit their receptors, essentially. So we should see an overall decrease in the amount of neuroinflammation in mice expressing less chemokine overall. So we needed to figure that out. Of course, right now we're just looking at neurons in a dish. We wanted to confirm that chemokine expression was actually diminished in vivo. And we did in fact see this. So using two different models of West Nile virus infection, either intracranial infection or the subcutaneous, the sort of peripheral route of infection, we saw a decreased expression of both CCL2 and CXCL10 in the brain following both of these infections. And this was particularly remarkable because chemokine expression tends to sort of track with viral burden. If you have a lot more virus, you usually see a lot more chemokines. So the fact that even though, remember, the RIP3 knockout mice have a lot more virus, we're actually seeing less chemokine. It really speaks to the importance of RIP3 and actually promoting the expression of these chemokines. So these differences are actually pretty dramatic in consideration of the virologic data. So, less chemokines is great, but does less chemokines actually mean fewer immune cells recruited into the nervous system? So we turn to flow cytometric analysis to answer this question. And so what you'll see is just very broadly, there's just a lot fewer immune cells in the brain of RIP3 knockout mice, but we'll dig in just a little bit here. So if we focus first on the T cell compartment, looking at CD4 and CD8 T cells, we see that mice lacking RIP3, both on a percentage and a total cell basis, have fewer CD4 T cells as well as CD8s. And in separate experiments, we also showed that these mice have fewer antigen West Nile specific CD8 cells as well using tetramer staining. So again, I think you can just sort of gloss over this and just see that the RIP3 knockout mice sort of just failed generally to recruit sufficient numbers of T cells into the brain following infection. In addition to T cells, we looked at myeloid cells. For those of you who have never worked in the brain before, remember that the brain has a large population of resident myeloid cells called microglia. So if we want to figure out whether there's a differential amount of recruitment of myeloid cells into the brain from the outside, we have to distinguish the resident cells that are already there from those that are trafficking in. So the simplest way to do that is to sort of gate on your myeloid cells, CD11b, and then stratify by CD45 expression. So microglia, resident myeloid cells of the brain, have a CD45 low or intermediate profile, whereas the infiltrating cells have a CD45 high profile. So if we sort of break it down this way and we look specifically at the CD45 high myeloid cells, we saw a decreased recruitment of inflammatory cells from the periphery, inflammatory myeloid cells from the periphery into the brain. Whereas if we look at the microglia, the resident cells, their numbers were equivalent between the genotypes. So this was a really nice, almost like an internal control. The resident population of cells was not impacted. There was no difference in microglial numbers. It was really the immune cells, the myeloid cells coming from the periphery that were impacted by the loss of RIP3. We sort of asked which myeloid cells this might be. And again, it was kind of a little bit of everything. Both sort of monocyte macrophage lineage cells expressing F480 were down, as well as dendritic cells, antigen-presenting cells expressing CD11C and MHC class II. So generally, what I hope you can appreciate is that RIP3 was really important for promoting a broad-based kind of neuroinflammatory profile in the brain of West Nile virus infected mice. When we lose RIP3, we lose a lot of really important immune cell subsets. Tying this back, though, to our chemokine data, remember that I introduced these two chemokines that were specifically downregulated in neurons and in the brain of neurons or mice lacking RIP3. So we should hypothesize in theory then that if we have decreased expression of CXCL10, we should see fewer CXCR3 expressing T cells, and similarly fewer CCR2 expressing myeloid cells when CCL2 is down. And we did, in fact, see exactly that. So if we look specifically at CXCR3 expressing CD4 T cells and similar CD8 T cells, we saw greatly decreased numbers of both of these populations expressing CXCR3. Similarly, we saw decreased CCR2 positive myeloid cells, the sort of F480 population of myeloid cells. So again, loss of these important chemokine axes really did bear out specifically in the loss of immune cells expressing their cognate receptors. So the way this kind of looks in summary is that if we have infection in a neuron, we get activation of RIP kinase signaling. And remember that instead of activating its canonical function, which is activating MLKL and necroptosis, instead RIP kinase signaling was driving an inflammatory transcriptional response, some of which included inflammatory chemokines. These chemokines were important for recruiting leukocytes out of the circulation into the infected nervous system, where they go about the work of controlling infection and ensuring survival of the host. So we're following up on this in multiple ways in my lab now. For one, we're really interested in how this pathway shapes neuroimmune responses, not only to infection, we're using other infection models, but also to non-infectious pathologies of the nervous system, things like neurodegeneration, Parkinson's disease, as well as traumatic injury, to see if there are sort of context-dependent roles for this pathway in promoting pathologic versus protective immunity. And then sort of one of my favorite questions is, does this pathway have different roles in other types of cells in the brain? We really focused on neurons in those two papers that I sort of introduced in the first part of the talk. But we're really interested in seeing whether or not non-neuronal resident cells of the brain also use this RIP kinase dependent pathway, in particular the cell death independent RIP kinase pathway, to shape neuroinflammatory responses to different diseases. So I'll close with a few slides on work that we're getting into now in my lab, which of course has been stymied a little bit by the pandemic, but we actually have made quite a bit of progress. What I want to talk to you about, again, just kind of closing here, is initial work that we're doing looking for roles for this pathway in cells of the nervous system known as astrocytes. So again, this is mostly immunologist, I think, and so I'll tell you a little bit about astrocytes. Most of you, I think, have a general understanding of neurons and what they do. Astrocytes are a population of cells in the brain that really support all the other cell types. They have really diverse functions, and for that reason, they're really my favorite type of cell in the nervous system. You know, they regulate neurons both metabolically and developmentally. They really shape neurotransmission at the synapse, so they have important neurologic functions, but they also have important immunologic functions. They sort of have a lot of intrinsic immunologic activity, and they also regulate the immune system through their regulatory capacity at the blood-brain barrier. And so you can sort of imagine astrocytes as kind of a bridge between neurons and the immune system, which is in the circulation. And so this kind of positioning of astrocytes within the overall, homeostatic biology of the nervous system, I think really makes them, a pivotal cell type in a lot of different disease states. And so for that reason, we're really interested in roles for astrocytes in all of our different disease models. And so some of the early questions that we're asking, of course, are just kind of starting, from the same place. Does RIP kinase signaling in astrocytes, is it important for the control of viral infection in the nervous system? And so to do this, we have a couple of cool animal systems in particular that I'll introduce you to. One is a conditional expression system where we can generate either astrocyte-specific knockouts of RIP3 or astrocyte-specific overexpressing RIP3 animals. And so these are sort of conventional cell type-specific knockouts using Cre-Lox. And if we infect these animals with intracranial Zika virus, we see that they are highly susceptible to infection. So it's not only neurons that matter, but you actually also need RIP3 in your astrocytes to control flavivirus infection. This result, which is very beautiful, which we're really excited about though, shows that if we actually add more RIP3 into astrocytes, so the mice have all their normal RIP3, but we just add a whole bunch more to their astrocytes and then we infect them with Zika virus, they actually do much better. Their survival rate actually improves pretty dramatically. And we see the same thing if we look at kind of clinical indices of infection with weight loss. Mice lacking RIP3 in astrocytes lose weight faster, they get a lot sicker, whereas mice with extra RIP3 in their astrocytes, they don't lose nearly as much weight, and overall they're much healthier. And so we are really interested in kind of, again, the mechanisms that are shaping this kind of top level difference in survival and clinical signs of disease. So far, we've seen, for example, that if we culture astrocytes or in vitro that are isolated from either wild type mice or mice lacking RIP3 or MLKL, we see that RIP3 knockout astrocytes have greatly increased replication of Zika virus in culture, while MLKL knockouts do not. So these data and others suggest to us that the astrocyte phenotype is also necroptosis independent. And we kind of see a similar thing if we grow human astrocytes, primary human astrocytes in culture, and we inhibit RIP3 with a pharmacological inhibitor. We also see increased replication of Zika virus. So suggesting that RIP3 has a cell intrinsic antiviral capacity in astrocytes. Sorry, I keep getting a little pop-up here. Okay. The last thing I want to introduce you to is another really cool mouse pharmacogenetic system that we're using. And here we have basically a version of RIP3 that is chimeric. So it's fused to a domain known as FV. What this FV domain allows us to do is to, when we express this in cells in vivo, in astrocytes in our context, we can add a small molecule ligand known as BB homodimerizer. This homodimerizer ligand binds with really high affinity to FV and nothing else. And so when you add the BB homodimerizer, you actually drive dimerization of this chimeric protein, which is sufficient to induce its activation. And when you do so, you can activate all of the downstream consequences of RIP3 activity, including inflammatory gene transcription. And so last data slide here, I know I'm running out of time, but in early experiments, we've shown very nicely that if we take mice expressing this inducible activation system of RIP3 in astrocytes, and we treat with BB using intracerebral ventricular administration, we see that mice expressing the construct in astrocytes, which is what this RxAC means here, really potently express a number of transcripts that are associated with inflammatory astrocyte activation. So we're really excited about using this model here in a non-infectious and a sterile system to actually answer really targeted questions about the molecular outcomes of RIP3 activation in astrocytes and other cell types in vivo, which this system will allow us to do. So right now we're working on further identifying the mechanisms of pathogenesis that sort of shape what we're seeing in our conditional expression systems. And then we're also really interested in doing transcriptomic profiling and other similar types of analyses on astrocytes using our different mouse genetic systems as well. Okay, with that, I'll thank my lab. It's been a year since we've been all together, so I kind of just had to find a face shot of everybody. But in particular, this work is really being spearheaded by a really great graduate student in my lab, Marissa Lindman. Our lab manager, Colin Atkins, has also been really important in this work. And then some former lab members also contributed to this, Sarija Ndukuri, an undergraduate student, and a rotation student, Kimberly Newman. And the published work, I'll thank my collaborators and of course my postdoc lab at the University of Washington. Okay, with that, I'll take any questions. Thanks.
Thanks so much for that, Brian. That was really, really nice. Really enjoyed that. We already have a question in the chat. Actually, we've got a couple of questions already. First one is, how does RIP3 dependent transcriptional activation interact with interferon?
This is a good question. It does seem that interferon can sort of shape the RIP3 transcriptional response. And this has actually mostly been studied in the context of necroptosis, though. So MLKL is actually a really potent ISG. The role of interferon actually in the cell death independent part, though, has actually not really been thoroughly studied. Certainly ISGs are included among the RIP3 dependent transcriptional response, but the extent to which like you need interferon as a stimulus to get that, we really haven't dug that deep into that. So I wish I had a more satisfactory answer, but they definitely seem to be interlinked, but we kind of haven't teased out all the details of that yet.
It's really interesting. Yeah, and then we've got another question, which is, would MLKL mice be expected to show less neuron bystander cell death in Zika virus? And have you looked at this?
So MLKL knockout mice definitely do. Well, okay, there are two parts to this. In Zika virus, I've never seen any effect of MLKL. So as far as I can tell, it's not that like there's parallel RIP3 activities happening where like necroptosis is happening, but it's just not relevant to disease pathogenesis. And then also the cell death independent part is happening. It really seems that necroptosis just does not get triggered at all by flavivirus infection. MLKL is expressed in the neurons and in other cells. It's certainly there, but it's not becoming activated. We have no idea why biochemically it doesn't get activated in this particular context. So for your specific question, mice lacking MLKL, they don't have any difference in immunopathology in Zika virus. However, in other models, particularly in like models of neurodegeneration and that kind of thing, MLKL actually does drive a lot of neuronal death. And so it really depends on which disease you're looking at and kind of what the input into the system is, which really determines kind of which outcome happens. And we're really interested in figuring out the mechanisms that shape that, but it's still really early so far for that.
Cool, thanks for that. We're going to take just one more question quickly before we move on. Do you want to?
Yeah, so amazing talk, really enjoyed that. I was just wondering if you've seen this sort of reduction in the number of recruited monocytes, T cells. It's the same across the brain or in different sections of the brain. And also, have you looked at choroid plexus or meninges, or have you seen any differences in terms of the composition of those compartments in your model?
No, actually, so this is one of my really intense interests, as you could probably tell, is I'm actually really interested in this. In the studies we did with West Nile virus and the knockouts, we did that on a whole brain basis, and we actually did not do a lot of imaging. So unfortunately, I don't really know if there was a regional basis. You know, we didn't see any regional differences, you know, or any clear regional differences in viral burden or in like chemokine expression or the transcriptional responses. So my guess is in this model system, there probably aren't major regional differences in immune cell recruitment either, although we didn't explicitly look. But you know, now we're kind of like, you know, I sort of moved from an immunology department. Now I'm back in home in a kind of a more neuroscience focused area. This is actually something I really want to follow up on, looking regionally, but also in the meninges and some of the other sort of portals of immune cell entry into the brain to sort of really think more carefully, kind of geographically within the nervous system, you know, where this is happening and thinking about why that matters to disease pathogenesis. But we really need to do a lot of that work before I can answer those questions. But yeah, thanks for asking that.
That's okay, thanks.
Yeah.
Okay, well, thank you so much. Like Juan just said there, that was just a really great talk. I really enjoyed that. Would you be able to just pop your social media details in the chat box? And
sure.
also, would you keep an eye on the question and answer box, just in case someone asks something there, and then you can type the answer, if that's okay?
Sure, I'd be happy to.
Oh, that's great. Thanks so much, Brian.
Great, thanks.
So with that, we are moving on to our next talk, which is from Mauro Gaya. And as a quick introduction, so Mauro is a junior group leader at the Center of Immunology in Marseille, France, and has been there since 2018, and became permanent at INSERM in 2019. And has been studying mechanisms regulating the initiation of B-cell immunity during infection. And with that, I will let you take it away.
Okay. Do you see the presentation mode or the whole screen?
No, we can see the whole screen.
Okay, perfect. Okay, thanks for the introduction and for inviting me to take part of this initiative. And I think I will be the first person in this seminar series to talk about B-cells. And in this case, how the innate branch of the immune system, such as the natural killer T cells or the macrophages, regulate the production of antibodies during viral and bacterial infection. So There. I'm a biologist from the University of Buenos Aires, and I did my master in plant biology at the Leluar Institute. And in 2011, I joined the lab of Facundo Batista at the London Research Institute to do my PhD. Now this institute doesn't exist anymore, and it's part of a bigger center called the Francis Crick. In 2016, I moved to Boston together with Facundo. And in 2018, I came back to Europe to open my lab at the Lumini Center for Immunology in Marseille. So this is the CIML Center. It's placed in a really nice area in France, in south of France. It's inside the national park. So here you can see this is my team right now. And this is the terrace of the institute where we have lunch with a really nice view of the Mont Puget, and this is like a half-an-hour walk from our center. So yeah, it's a really nice place to work, not only scientifically, but also nature-wise. So we all know that vaccination is the best achievement of modern medicine to protect us from some deadly pathogens. And as you can see here in this number, these are the number of cases in the US before and after vaccination. As you can see, the number of cases dropped dramatically. And in some cases, we even managed to completely eradicate some diseases, like in the case of smallpox or polio. And hopefully next year, we could add the SARS-CoV-2 vaccine into this list. One of the things we know about vaccines is that the success of a vaccine is associated with induction of high titers of antibodies. And these antibodies are produced by B cells. And despite the importance of B cells in vaccination, it was not really clear until a decade ago how B cells were encountering the pathogens that were entering into our body. However, these three groups, the group of Ulrich von Andrian, the group of Jason Sister, and the group of Facundo Batista, what they found was that when pathogens enter into the lymph nodes, they are trapped by a layer of macrophages at the subcapsular sinus. And then B cells that they are found in these underlying B cell follicles, they can trap the pathogens from their surface. In fact, we can visualize this by 2-photon microscopy. Here you can see the B cells in red that they are trapping the antigenic bits on the surface of the macrophages. And after that, they move to the borders of the follicles where they present peptides derived from the pathogen to the T cells. The T cells give help to the B cells in order to differentiate into plasma cells or enter into the germinal centers where their affinity will increase to finally produce high affinity antibodies. So one of the first things we notice after when we have an infection going on in our body is that our lymph nodes get bigger. And this increase in the size of the lymph nodes goes together with a complete remodeling of the lymph node architecture. So we wanted to know whether infection has any impact on the organization of this macrophage barrier at the lymph nodes. So to answer this question, we did a really simple experiment. We took some wild-type mice, we infect them with influenza virus intranasally, and after 6 days, we harvest the lymph nodes draining from the lungs, that are the mediastinal ones. This is a confocal section of control lymph nodes, and as you can see, there is a continuous layer of macrophages in green that they are stained for CD169, and the B cell follicles that are stained for B220. So in mice that were infected with influenza, we see an increase in the size of this lymph node, an increase in the follicle, but more surprisingly, we see a complete disruption of this macrophage barrier. And in fact, this was not specific for a viral infection, but also we observe the same if we do a bacterial infection, for example, in this case with Staphylococcus aureus in the ER, and we harvest the cervical lymph nodes, a complete macrophage barrier disruption. And it's worth to mention, I wrote it here because I always forget to mention it. This is something that is localized, so it happens only on the draining lymph nodes, but not in the lymph nodes, on the rest of the lymph nodes from the body. And also it's a temporary process. After more or less 1 month, all the macrophage barrier remains, goes back to normal. We then wanted to know whether infection was really necessary to induce this process or whether an inflammation per se could induce something similar. So for this, we inject CpG in the foot pad, and then we harvest the popliteal lymph nodes. And as you can see, we observe a similar phenomenon. This is 2-photon imaging of the lymph node capsule, and this is a side view. And as you can see, in the steady state, we see the collagen capsule and the macrophages all at the subcapsular sinus. And after yes, inflammation, there is a migration and or a retraction of macrophages from the subcapsular sinus to the inner areas of the lymph node. We then went into the mechanism inducing this macrophage reorganization. We thought it could be mediated by macrophage pyroptosis, or by cell killing mediated by neutrophils and NK cells, or by signals provided by B cells or T cells. However, so we analyzed a lot of knockout mice that were defective into these different pathways, but we didn't observe any difference at the level of macrophage disruption. However, what we observed was that after inflammation, there is a big recruitment of migratory dendritic cells into the subcapsular sinus. And as you can see here, it looks somehow like the dendritic cells arriving into the lymph node are somehow pushing away the macrophages from the subcapsular sinus. So our theory was that when there is an inflammation, then you have a wave of dendritic cells arriving into the lymph node, and this dendritic cell will somehow push the macrophages away from the subcapsular sinus area. If this is true, then if we block the entry or the migration of the dendritic cells into the lymph nodes, then the macrophage disruption should be reduced. So for this, we use CCR7 knockout mice where dendritic cells do not migrate into the lymph node. And surprisingly, what we saw is that even if in the lymph nodes of these mice, even though they are increased in size, the macrophage barrier remains intact, suggesting that dendritic cell migration is necessary for the induction of macrophage disruption. Then, as an alternative, we also block the sensing of the inflammation by dendritic cells using a MID88 flox CD11-secrete mouse. And in that case, we also observe the same, an increase in the size of the lymph node, but the macrophages are still there, suggesting that dendritic cell migration is needed to induce this macrophage barrier disruption. So we wanted to know which is the physiological role of this process, of this phenomenon. So until now, we show that both viral, bacterial infection, and even CPG or inflammation can induce macrophage barrier disruption. And this is something temporary. So what happens if during this period of time where the macrophages are not there, a second pathogen arrives into the lymph node? Can it be captured by the subcapsular sinus macrophages? And in this case, we observe that this is not the case. This is here you can see that we injected fluorescent beads in mouse that didn't have a primary infection going on, and you can see the accumulation of the beads on the subcapsular sinus. However, if there is a primary infection and a remodeling of the macrophage barrier, the antigen is not trapped, cannot be kept at the subcapsular sinus. Then can B cells get access to this secondary antigen if the macrophage are not there? And in fact, what you can see is that in the case when there is no primary infection and the macrophage barrier is intact, you can see the antigen-specific B cells with the antigen inside. However, in the case of when there is a primary infection ongoing, then the B cells cannot capture the second antigen. And in consequence, what we see is that B cells, they are not able to form, in these cases, plasma cells or even germinal centers. So at the end, we came to this conclusion, is that we propose this model that during a primary infection, the pathogen arrives into the lymph node, it's trapped by the macrophage barrier, and B cells can recognize the pathogens there and start producing antibodies. However, at the same time, there is a wave of dendritic cells arriving into the lymph node that bring antigen and present it to the T cells. And these dendritic cells, when they arrive, somehow they disrupt this macrophage barrier. So what could be the evolutionary or physiological advantage of this phenomenon? This we don't know. So we hypothesize that this macrophage retraction could somehow help the entry of immune cells directly through the subcapsular sinus floor. It could also facilitate the antigen transport by the macrophages to the deeper areas of the lymph node, or also the release of pro-inflammatory cytokines also into the deep lymph nodes. And while all these scenarios might maximize the primary response, this is at the cost of leaving the lymph node temporarily unable to respond to further antigenic challenge and for the production of antibodies against this second pathogen. So what we believe is that here we propose a mechanism of why, when there is a primary infection going on in our body, like in the case of influenza, we are more susceptible to get, for example, a secondary bacterial pneumonia. And also we think this can give us some clues for vaccination, because as I told you, this is something, a localized process. So for example, if we receive a vaccine on one arm, at one point, this induces the remodeling of the lymph node when there is an immune response. So then if we will receive a second vaccine during a short period of time, right after the first one, then maybe this vaccine should be given on the other arm in order not to avoid the generation of antibodies against the second challenge. So then now I'm going to move away to talk about some non-canonical ways of B cell activation. So we know that the normal way that the B cells get activated is by when they recognize pathogens, they present peptides to the T cells, and then they produce antibodies. However, there are some pathogens, like in the case of Sphingomonas, Borrelia, or Streptococcus pneumoniae, that they have glycosphingolipids on the surface. And in fact, when these pathogens are internalized by the B cells, B cells can present these glycosphingolipids on the surface in the context of an MHC-related molecule that is called CD1D. And then there is a specific population of T cells that they are called the natural killer T cells, that they have a TCR specific for lipids. So then in that case, NK T cells can give cognate help to the B cells in order to enhance the antibody production. So for example, in the case of these pathogens, B cells will internalize this pathogen, they will present the peptides to the T cells, the lipids to the NK T cells, and both of them will enhance antibody production. But the truth is that these glycosphingolipids are not really abundant in nature. Like only some special kinds of bacteria have these glycosphingolipids. So then we wonder whether NK T cells would play any role in the B cell physiology, even in the absence of these glycosphingolipids. So to address this, we use, again, the influenza virus model, because we know that viruses don't have glycosphingolipids. So we infected wild-type and NK T cell deficient mice with influenza, and then we harvest the lung-draining lymph nodes at day 9 of infection. As you can, then we check germinal center formation. This we check it by the upregulation of FAS and GL7. And what you can see in the wild type, we can see the formation of this really nice germinal center population compared to the uninfected mouse. However, in mice that do not have NK T cells, there is a dramatic decrease in the germinal center formation. And when we do some imaging, we found that this decrease in germinal center is both because of there is less germinal center structures compared to the wild type, and they are also reduced in size. So we wanted to know how NK T cells were providing help to the B cells to form or to seed these germinal center structures. So our first hypothesis was that B cells, upon sensing of infection or upon sensing of the pathogen, they will induce the presentation of lysosomal or some endogenous lipids, that this would be presented to the NK T cells, and NK T cells could provide cognate help to the B cells, even in the absence of exogenous glycolipids. And it was not just a crazy idea that we thought about, but in fact, this is the case of what dendritic cells do. Dendritic cells, for example, if you stimulate with CpG or with LPS, they can start presenting lipids from the lysosome to the NK T cells, and NK T cells can help the disease. So to see whether the cognate interaction between B cells and NK T cells are required for germinal center formation, then we wanted to block this interaction. And if that is the case, then if we block this interaction, we should see a reduction in the GC formation. So to block this interaction, we generated a CD1D flux mouse that we crossed it with a mouse expressing the Cre recombinase under the promoter of MB1. So as you can see, this mouse model works really well and is really specific. So here you can see CD1D expression only on B cells, and the Cre negative mice, CD1D is expressed. Cre positive, there is a 100% deletion of CD1D on the B cells. So then we went on to infect Cre positive and Cre negative animals with influenza, and we harvest the lymph nodes draining from the lung at day 9. And surprisingly, we didn't see any difference at the germinal center formation. So we were quite a bit disappointed about that, because this was like all the project was built on these endogenous ligands and cognate interaction between B cells and NK T cells. But then we continue checking how if NKT cells are not helping the B cells through a cognate interaction, is it possible that they are providing soluble mediators to the B cells to see the germinal centers? So we went on to measure NKT cell activation, and the way that to recognize NKT cells is because they are positive for TCR beta, and they can also bind to a CD1D tetramer. As we can see, the peak of NKT cell activation is around day 3, measured by CD69. And we can also we measure the cytokine production by day 3, and we can see that NKT cells can produce interferon gamma and interleukin 4 more or less at the same level. However, something really interesting happens if we do a reverse gating. Instead of gating of how many NKT cells produce interferon gamma, we gate of all the cells producing interferon gamma in the lymph node, how many of them are NKT cells. We found that less than 1%, indicating that having 1% more or 1% less of cells producing interferon gamma won't affect much the response. However, if we check of all the cells producing IL-4 in the lymph node, that is much less than the ones producing interferon gamma, how many of them are NKT cells? We found that around 60% of the IL-4 producing cells in the lymph node are NKT cells, suggesting that NKTs are the main source of IL-4 in the lymph nodes. And this was surprising because at this point, these papers that I cite here, they were saying that most, and one of them says that all the IL-4 produced in the lymph nodes after infection is coming from T follicular helper cells. But T follicular helper cells appear quite late, later in the immune response. So then we went on to analyze the contribution to the IL-4 production by the natural killer T cells and by the T follicular helper cells. So for this, we infected the mice with influenza. We harvested the lymph nodes at different days of infection. We gated on the cells producing IL-4, and then we checked NKT versus TFH. As you can see, by day 3 of infection, around 70% of the cells producing IL-4 were NKT cells. This trend starts to be reversed by day 6, and by day 9 of infection, most of the IL-4 producing cells are T follicular helper cells. So this indicates that there are 2 waves of IL-4 production, an early one that is produced by the NKT cells, and a late one that is produced by the T follicular helper cells. So we know that the T follicular helper cells, they produce IL-4 in the germinal centers. So we wanted to know where the NKT cells were producing the IL-4. So we want to visualize NKT cells doing in situ microscopy. Here you can see that NKT cells are stained here for tetramer, are very abundant in the follicles, in the B cell follicles in the lymph nodes, but also in the interfollicular areas. So to see where the IL-4 was being produced, we took advantage of an IL-4 GFP transgenic mouse. And as you can see, this was quite surprising. None of the NKT cells that were found in the follicles were GFP positive. However, most of the IL-4 positive NKT cells were found in the interfollicular areas. Then I'm presenting this. This was some of the things that the reviewers were asking us to do. Like they wanted to know, okay, 60–70% of the cells producing IL-4 were NKT cells. What are the other ones? Are they ILC2s? So in fact, what we saw is like we did single cell RNA-seq on the IL-4 GFP positive cells. Around 70% of them were NKT cells, and of the rest, we found that these were not ILC2s. In fact, ILC2s, they didn't contribute to any of the IL-4 production in the lymph nodes. What we found was that the NKT cells, there were 2 populations of NKT cells, one's ROR gamma T positive and the other one TBET positive, and the ones that were non-NKT cells were mainly T cells. We found the population expressing CD4 and another one expressing CD8. And this is what you can see here. The 4 populations are CD3 positive. The NKT cell populations that are these 2, they express a PLZF, which is a transcription factor necessary for NKT cell development. One expressing TBET, the other one ROR gamma T, but both of the NKT cell population expressing GATA3, which this is something really typical of natural killer T cells. Like in the case of conventional T cells, you find that they express TBET, they express ROR gamma T, or they express GATA3, but not at the same time. But NKT cells can express these transcription factors together. But the interesting thing was that one of these populations, the TBET positive one, was expressing very high levels of CXCR3. And why this caught the attention was that because CXCR3 was the chemokine receptor involved in the localization of conventional T cells in the interfollicular areas. That is where we saw the IL-4 production. So then what we did was to sort CXCR3 positive and CXCR3 negative NK T cells and check for the IL-4 production by ELISpot. And as you can see, the main source of IL-4 is coming from the CXCR3 positive NK T cells that are the ones placed at the interfollicular areas. So this is a bit of the summary. We have the early NK T cell wave of IL-4 produced at the follicular border. So then we wanted to know whether this early wave of IL-4 is somehow necessary for the seeding of the germinal centers by the B cells. So for the first thing we did was to use an IL-4 knockout mouse, and we saw a dramatic decrease in the germinal center response. However, in an IL-4 knockout mouse, we were blocking both the early and the late IL-4 waves. So to block only the early one, we took wild-type mice and we administered an anti-IL-4 antibody during the first 3 days of infection. And what we see is that in mice where we block the early wave of IL-4, we see a reduction in the germinal center response. And then we did the reverse experiment. What happened if to mice that do not have NK T cells, we give IL-4 only during the first 3 days? And we found that giving IL-4 only during this early window of time is sufficient to recover the germinal center response and to induce it even to higher levels than the one that we observe in wild-type mice, suggesting that IL-4 is a potent inducer of germinal center formation. So to summarize, what we describe is that when there is an infection, the pathogens are tried by the subcapsular sinus macrophages, and then B cells move to the follicular borders. And these same macrophages that activate B cells can also activate NK T cells through a mechanism involving CD1D and IL-18 that I didn't show here. And then these CXCR3 positive NK T cells that are found in the interfollicular borders produce an early wave of IL-4 that is required for the B cells to go back to the follicles, form germinal centers, and induce the production of antibodies, and in particular of IgG1 antibodies. So we were the first one showing that there is an early innate link between the NK T cells and the B cells involving IL-4. And I would like to thank this study was performed it was supported by the Cancer Research UK, by the Francis Crick Institute, and by the Oregon Institute. For the first study was mostly done in London. So I would like to thank the lab of Facundo Batista in London and our collaborators there, Caetano Reise Sousa, David Withers, and Antal Roth. And the second study that was mostly done in Boston. So this is the team in Boston. And our collaborators for the second study that were Patricia Barral, Daniel Lingwood, Alex Shalek, Dan Baruch, Mark Wilson, Jessica Street, and Padraig Fallon. Thanks to you for hearing, and hopefully I will be able to present what we are doing in Marseille here. Now it's a bit too early because we are just starting to develop the projects, but in a future edition of these talks, I will be able to start showing what we are doing here. So thanks a lot.
Thanks very much for that. That was a fantastic talk. I really enjoyed that. If anyone has any questions, you can write them in the question and answer box. One thing that I was wondering, just while people are writing questions, so obviously you saw the change in the macrophage barrier in the lymph nodes during viral infection and salmonella infection. Would you expect also in the draining lymph nodes to see that, for example, in helminth infection or in cancer, for example?
Yes. In fact, in helminth infection, I don't know. Like nobody has shown that. And in cancer, there was a paper coming out after hours where they were showing that they were checking the lymph nodes where there was metastasis and in humans, and they were seeing as the same macrophage reorganization that we saw after viral and bacterial infection. Okay, that's really interesting.
That's really interesting. So we have a question from, I don't know who from, an anonymous attendee. Is influx of macrophages necessary to clear out debris resulting from cells that don't pass the affinity test when searching for specific antibodies?
I don't understand much the question. Is an influx of macrophages necessary to clear out debris? So in fact, these macrophage I don't know if I'm answering the question that he's asking or she's asking, but so if it is necessary to clear out the debris, so in fact, these macrophages, they are poorly phagocytic, or they are there, like, that's why, like, this one of the characteristics of these subcapsular sinus macrophages, that is when pathogens are there, they are not internalized by these macrophages. In fact, they are retained there and the B cells trap it from their surface. So I don't think that these macrophages will be the ones that will clear out debris or other pathogens.
Okay.
Was it this what the person was asking?
I mean, I think so.
Okay.
That would be my take on it. Carlos is asking, do you have any idea what signals move macrophages to the inner part of the lymph nodes?
So for this, we did a lot of experiments and all of them came negative. What I believe is that this is not just a physical movement, because what, the dendritic cells, when they enter into the lymph node, they enter mostly through the interfollicular areas. So if it will be just a physical pushing of the macrophages, then we should see more disruption at these areas than in the other ones. And in fact, we see a very uniformous disruption. So what I think is that dendritic cells are somehow producing a soluble mediator that could somehow release the macrophages from being there. Like we were thinking at some point, we know that the macrophages are attached to the extracellular matrix by, how do you say? Attached to the extracellular matrix. So then we wanted to see whether, and we know also that dendritic cells, they produce metalloprotease after activation. So we thought at some point that maybe this metalloprotease produced by dendritic cells could somehow degrade a bit this matrix in the subcapsular sinus and inducing the release of the macrophages. But this is just an hypothesis and we didn't test that.
That's really interesting. Yeah, it's really, really cool. Well, if nobody has any other questions at the moment, I'd just like to say thank you very much again for a really great talk. Would you also be able to just pop your social media details in the chat box so people can contact you if they have any questions?
Yes.
So that's the end of the session for today. So thank you again to both speakers for great talks. Next week, we are moving 1 hour later because one of our speakers is joining us from New Zealand. So next week we have Kevin Ng from the Francis Crick Institute, and we also have Francesco Vacca, who is in the Malagan Institute of Medical Research, and previously at the University of Edinburgh. And then next week, we also have an additional session on Wednesday at 4 p.m., which is from Paul Mackay, who is in Robin Shattock's lab at Imperial College and has worked on COVID-19 mRNA vaccine, which is currently in phase 2 trials. So join us for that if you can as well. And with that, just thank you again for coming along and hope to see you all soon. Thanks. Bye.
Thank you.